Right, so I'll be just talking about the FATHMM family of predictors, and I should first of all want to start actually just mentioning the main players here. Hash Shabab is in the audience here and played a major role throughout development of these predictors, and Mark Rogers also here, particularly with data integration. And then there are various slobs like me who can make vague suggestions to people, and sometimes our ideas work. Right, so there's been several papers. The first one's really to do with sequence conservation. Quite obviously, if you've got variants in regions which are highly conserved across species, much more likely that the hit there is pathogenic than a hit in a region where you've got quite a lot of variability across species. And then, of course, if you make your predictors disease-specific, maybe cancer or other diseases, then they're more accurate still. However, the paper I want to talk about is perhaps this latter one, which is to include data integration. So you'd be using the idea of sequence conservation, yes, but you'd use as many different types of data as you possibly can. My own interest actually is, I come actually from the machine learning area rather than bioinformatics strictly, and so in the past I've been actually cooking up some of these sort of data integration algorithms, so that's sort of how I wandered into the area. But other interest, of course, was to build a predictor in non-coding regions, which was just discussed with Graham Ritchie, because at the time we considered this, there wasn't any predictor there. So, of course, so we're interested both in coding and non-coding regions, and of course our already existing algorithms. But this interest in non-coding variants is because the majority of catalogued disease-associated variants fall in regions which are in non-coding regions, intronic or intergenic. So in our study, we are using a pathogenic data set already mentioned, this human gene mutation database, and for the normals, but we're looking at them again in a second, the 1,000 Genomes Project data. And there was some filtering involved. I did say the interest here was to actually devise predictors based on other types of data beyond sequence conservation. So this involves data integration, and the 10 I'm mentioning here, which I'll call feature groups, was our starting point, though we've actually been examining other types of data as well. So what you want to do at the end of the day is build a predictor that uses different types of data. If you want to do data integration with supervised learning, there's two sort of ways you could sort of do it. One is you could build a classifier for each individual type of data, and then in some way combine your classifiers together, probably in a weighted scheme where you give greater weights to those subclassifiers or base classifiers which are more accurate. So you might do it at that level. However, in this talk, we're doing it more at the data level, which is using multiple kernel learning. So the idea of multiple kernel learning is you take these different types of data, you construct a kernel matrix, and the kernel matrix represents the similarity or dissimilarity of data objects. And you can now derive kernels for all different types of data, discrete data, continuous data, graph kernels, sequence kernels, all these sorts of things. So you can take any type of data, perhaps this, put it into a kernel, and with multiple kernel learning, you're doing two things. You're wanting to find a learning parameter, but also the kernel weights, those coefficients in front of the kernel matrices, which go up and down depending on the significance of that type of data. There's around about 25 or 30 algorithms actually proposed for multiple kernel learning, and they fall broadly into two classes, depending on the regularization involved in the kernel weights. So at one end, you may be rather sparse with kernel weights. If a type of data doesn't look significant, it might give you 0 as the kernel weight. And then there's perhaps denser combinations, which tends to use all the data and so on. So you've got a variety of multiple kernel learning algorithms. So this is the algorithm FATHMM MKL. So these are different types of data you may want to encode into kernels and put into the classifier. It doesn't matter how many types of data you use, because in fact, if the data is irrelevant to the problem you're considering and your algorithm's good, it'll zero weight that particular type of data. In fact, you can create kernels from random data just to check out your algorithm, and it should zero weight that particular type of data. It was just rubbish. So those are the 10 types of data we're considering. And it turns out the story for non-coding is different from coding. Whereas with coding, many more types of data are relevant to the prediction. Indeed, all those 10 feature groups were players. With non-coding, less available, useful information. And it turned out that 4 feature groups was the model we were considering for non-coding regions. These are our results for non-coding. There's many issues I'm sort of skipping over. One is that you have to avoid bias with these predictors, because if it's a case that in some way the same example pops up in the training set as in a test set, it makes it tremendously easy for your classifier to classify something it's already learned. So there's all those sorts of issues, but in the context of multiple kernel learning, it's more dangerous, because if one of those types of data actually has that sort of problem, then your algorithm would give it extra weight, because after all, it's working pretty well. So you have to really check your data very carefully. You don't have such bias or circularity involved. But here we're talking about non-coding and the various ROC curves, and the FATHMM MKL, which is used in these 4 different types of data. Its performance is in black here at the top. Now, sequence conservation does work pretty well by itself, and it's the red curve. And then these other types of data, they contribute to some extent and lift your performance a little bit. So that is the FATHMM MKL. There's now FATHMM MKL2 that Mark Rogers was talking about yesterday, which is a bit better. But this is a comparison with Guava, which was just discussed, and CAD. And so it seems like FATHMM MKL seems to be better on this HGMD 1,000 genomes data set. These were the kernel weights I was mentioning just now. As you see, sequence conservation gives greatest weight. Well, I've got a lot more to do. Oh, you gave me 20 minutes, didn't you? On your emails. Oh, dear. Right, so these are the kernel weights. You can introduce a probability measure on top. And we were interested in doing this because we want to understand, in an experimental context, a biologist could be interested in your highest confidence predictions. So what happens when you restrict yourself to highest confidence, and naturally your accuracy on this reduced set increases, near enough to probability of 1, and the proportion, though, you're making predictions on decreases. Right, so I've got limited time. Coding, 10 different types of data relevant, and again, if you string them together, multiple kernel learning, you get this sort of performance. This has since been improved, so that's that. So this is a summary of where we are, and let me just say a few other things. Is the data clean that we're using? Well, one way you could actually consider that question is common variants are unlikely to be associated with disease. Evolution will get rid of them. Variants would, on the whole, be rare variants. Also, a common variant would give us a little bit of a bias with your predictor, because being common, there could be some sort of bias that's more likely to classify those correctly. So let's just assume, though, that without loss of argument here, that perhaps rare variants, common variants should be predicted equally well. Well, we don't observe that. The apparent false positives are very much concentrated here in this region as against neutrals. In other words, it appears to be making mistakes predominantly with rare variants in terms of false positives. So that's an indirect insight suggesting that in fact your negatives do contain actually a number of what should be positives. The data is not clean, in fact, the correct view is really negatives are unannotated, not really that they're unassociated with disease. Now, I'm down probably about 1 minute. So it works pretty well against CAD. And right, just checked it on another type of data, ClinVar data set, and also extra data that have been added in to HGMD database while we were sort of doing all of this. So performance in ClinVar, sort of effectively unseen data, works well, okay, and is competitive.
Coding regions works well and is competitive. We also did try on this unseen, apart from ClinVar, unseen HGMD data, and again, it works quite well there, 88%. So it does seem to be working well on unseen data. Finally, you can put all of these predictors into different tracks in a genome tolerance browser here, which Hash has been looking at. These are polyphen and SIF. They only predict in coding regions. And then there's these other algorithms. I'll not put all the tracks in there. These are just randomly picked. This is FATHMM MKL. And so you get some sort of curious insights here. You can spend hours just browsing around the genome and trying to figure out what it all means. But these strange little areas, this is just a randomly picked non-coding region. And these regions where you get peaks of one, no gene in that area, which apparently are significant. So that's your, it's not junk DNA out there, it's something or other. Right, so just to wrap up, there are many different classifiers you could do, there are many different multiple kernel learning algorithms, many things you could try, and other types of data you might want to play with. I think we're playing with at least 20, but there's probably more data you could use. There's indels as well. And just sort of moving on to our discussion of later, I just talked about the regions of interest with the genome tolerance browser. We've signed up to this Genomics England 100,000 genomes project. So we're thinking about some sort of pipeline where these various tools are used and a genome comes in, or the other interesting problem of multiple genomes come in, perhaps prostate cancer, and what comes out the other side are a bunch of files giving you information from these different predictors, perhaps not just sequence variants, but also indels as well. Other people probably doing the same thing. Well, I'm probably out of time. And we're hoping maybe with perhaps a vet team and others to construct with others a sort of pipeline with all these predictors in there. And I should shut up at that point.
Thank you.